All right, so welcome to this ever first episode of Vibrant's Map of the Month, which is April 2026. And our guest today, and also the guy who contributed the map, is Peter Gordon
Hello.
from UCL. Yeah. So, and maybe Peter, maybe you can give like a short introduction to yourself.
Yep. So I'm Peter Gordon. That bit is true. I work at the Sainsbury Wellcome Centre, which is linked with UCL, and I work as a senior microscopy specialist, sort of like a staff scientist in the imaging facility. So there are normal tasks like the training and maintenance of microscopes, but luckily for me, about 90% of my time is developing sort of wet lab techniques for the people of the building. One of them, which is relevant to today, is tissue clearing and antibody labeling of targets, so that then I can give the protocol out to the building, and then this becomes something that's done as a sort of a service. The entire building, I guess, a bit of context, is all around circuitry. So we do so many full mouse brain and rat brain imaging and crab brain. There's some weird stuff that happens here. It's not just mouse. But the idea is that we're looking at kind of a more of a holistic approach to looking at the brain rather than your standard histology. So that's a bit of a specialism that we have here. We have light sheet, mesoSPIM, and we also have 3 serial section two-photons, which people might know as brain saw. My boss Rob makes them. So we are a bit specialized in full brain labeling, imaging at least, and now labeling, hopefully.
Yeah, cool. So, and I think so. I mean, for the purpose of the talk today, so obviously the map that we are showing this month is TH staining in the brain, and maybe you want to explain a little bit about how just how you got started with this, and where it is.
So the TH. So we have. There's multiple parts to it, but the base for the TH, I guess, would be that there are groups in our building that are interested in Parkinson's, and tyrosine hydroxylase is very important in making dopamine. Dysfunction of TH and dopamine levels is linked with Parkinson's, and this group wanted to look at different animal models of Parkinson's, but looking at different areas within the brain and how the TH levels are different. That's the kind of the base of the TH labeling, because they're interested in different areas, and the TH labeling is sort of lovely and throughout the brain, lots of projections and cells and interesting different shapes. That's why it made sense to look at the full brain. Basically what we did then was, well, we did the tissue clearing at labeling, and then we imaged it on our mesoSPIM, but we also then imaged it on our brain saws. So that same brain was washed back from being transparent, run on our brain saw, because that then allows for very easy fitting into the Allen Atlas, and also the different tools that we have here, like Cell Finder and the different software packages that are developed in-house. So it all kind of fit into that pipeline that was already in place. So quite a versatile method we can run on different microscopes. The other part of it is, in general, we would like to move into different targets like CFOS, like perhaps using antibodies or nanobodies to boost GFP or mCherry signal when people are looking at viral projections, because when you're looking at these things in nice thin slices, you need to do immuno to really boost these fine fibers out. So what if you could do that in a full brain on the light sheet or our brain saw to do full brain axon tracing? So there's a lot of lead on from the TH. So it was a nice initial test. And also, obviously, it's the antibody comes from Miltenyi, and there is a lovely recommended concentration, which is very important in labeling full brains or tissues, because it saves months. And the protocol, at the same time, the protocol that you're giving to people, the Vibraint protocol, worked perfectly. So it was a lovely combination of these two things that resulted in a lovely picture, basically.
Yeah, and I think, I mean, obviously, TH is one of my favorite stainings. It's just so beautiful when you
It's really
see
cool.
it. Yeah. So, and I guess it's the balance between, you have these cells in the middle of the brain that kind of projects to both places, both the hindbrain and the front brain, and so it's like, it just looks really, really good.
It's a good argument for imaging the entire brain. There's these huge, lovely projections everywhere. You need to get them all.
It's the argument for doing whole brain imaging. You can say, So I think so you said you imaged on a mesoSPIM. So how, at kind of what resolution and step size did you end up using?
Ooh, that's a good question. So resolution, our mesoSPIM is generally for lower resolution stuff. So we would be working with pixel sizes of, let's say our Voxel would be say 6 by 6 by 5 in Z. We generally don't go lower than 5 in the Z because that's roughly what the light sheet thickness is. There's a lot of dependencies when it comes to the light sheet. But yes, roughly that's probably what we'd use it for. Whereas if we were wanting cellular or slightly subcellular resolution, that's when our brain saw would come in because it uses a 16x objective. So we can use it. It depends on the question. The light sheet is obviously allows you to image it. If you get the settings wrong, re-image it, reuse the brain because it's non-destructive, whereas our brain saws are effectively a vibratome and a multi-photon that chop an image and chop an image. So if you get the settings wrong, there's no more brain.
So, and I think one of the things you also mentioned, so obviously when you do the light sheet, you clear the tissue, you deform the brain essentially, right? So, and I think this is where, can you combine the data you get from the light sheet? You said, and you take the light sheet data, and then you take the same brain, you do the chainsaw cutting with its multifocal. But can you combine the data sets afterwards? Is that possible?
It's possible. I've not done it. So in-house we have Adam Tyson runs the neuroinformatics unit, which are essentially specialists in atlas making and creating, say, templates and warping different brains imaged from different modalities into the atlas space, and that would be the case here. So I guess when you're using the different clearing methods, you're right. If you use a solvent-based method, it'll shrink, hopefully uniformly. And if you're using, say, something like cubic, an aqueous method, it'll expand uniformly, hopefully. So with this, I tried iDISCO, and I also tried EasyClear, which I have found changes the shape of the brain the least. But when there will be more of these brains run, the idea would be that we could hand off this data to our NIU team that would then fit it to an atlas, either by creating a template based around the clearing method, so like iDISCO-based brains or EasyClear brains, or creating like a warp field that allows you to modify it because you've got that same brain on the brain saw in its natural state, and then the perhaps slightly shrunken or expanded version, and you create kind of like a warp template to just allow them to line up. So having those two data sets is super useful, and that would be something we do in the future.
Yeah. And I mean, obviously that's very close to my heart also, because that is kind of the whole foundation of Vibraint, is that we want to use this. I mean, we call it integrative spatial biology, in the idea that you can integrate multiple data sets into the same template and bridge data from one template to another template. And I think this opens up for so many possibilities also when you start talking spatial transcriptomics and all of these other things.
It's true. Like even if you go to that same brain can then be sliced in a vibratome. So say you actually find out in particular regions you need the kind of resolution that a times 40 or times 60 can bring. That brain can then, that regions can be sliced, imaged in super high, well, it's not super resolution, very high resolution, and then using, again, registration tools, align that to the atlas, to that brain, and you can kind of combine all the data sets in one big, lovely brain blob.
Exactly.
So it's super flexible.
Cool. So next up, it's like, I think the idea of this series is also maybe to a little bit also talk about some of the challenges around, iDISCO labeling. And, I think one of the things everybody who's doing iDISCO experience is that they're wasting a lot of time optimizing antibodies and things that are optimizing protocols. So one of the things I wanted to ask you is like, when doing this studying and getting started doing TH labeling, is there anything like some learnings you had on the way that you could reflect
I guess
on?
yes, in general, if you're trying to do full brain antibody labeling, you can potentially lose months. So it perhaps takes about a month to figure out if your concentration was wrong. If you're using a primary and a secondary, those are then two antibody concentrations you have to optimize together, and it's expensive because you're using quite a lot of antibody for the brain, and kind of. That's an overall challenge of full brain labeling. Just be aware it's going to take a lot of time, potentially. The reason it didn't actually take much time and worked first time for me, which is, I assume it will always work first time for me now, is it's the combination of, obviously, the protocol that you provided that you have run many, many times and know that this works, but it is the production of directly conjugated primaries that are now proven to work in either tissue clearing, so there's like iDISCO filters on company websites, or they are proven to work as 3D immunofluorescence and give you a starting concentration that works. And that's where you save so much time. The other part, it's a little bit more specific to iDISCO, is overall, as a protocol, it's not hard, but there are a lot of liquid changes, and it's tedious-ish. So there's a lot of ramping up and ramping down through methanol and different chemicals, and when it's an hour per like chemical change, it's a bit of a time sink. It's hard to kind of go away, do some stuff, come back, change that. I mean, it's not a huge problem. It depends how busy you are. The other thing that, again, if I had time, you can create an automated clearing machine just with some Arduinos and some pumps, which are sitting on my desk for when I have time again, so that part is now automated. But yeah, it's a hard one because I got it working first time. But basically, that's because there are now antibodies coming out at a recommended concentration that are directly conjugated and bright, so you only need to verify one of them. It suddenly makes everything a whole lot faster and easier. Less soul destroying.
Yeah, exactly. I mean, that's also very much our experience right now, that going from primary and secondary and then going to directly conjugated, really, it does, maybe you lose a little bit of signal intensity, but I think the overall signal to noise is kind of maintained.
Yes.
When not adding the antibody, secondary antibody usually also introduces some kind of noise in the system.
Yeah.
That you then
secondaries are normally polyclonal and they're a very high concentration.
Yeah, and sometimes they give some precipitation, like you see small precipitates in the brain and small speckles and stuff that are just
clumps.
Even if you have no primary, they will still provide some kind of low background signal. So I think in our hands, like when the primary conjugation works, you get so much better signal quality. So that's also, it's not always possible to do that. I think many of so of course Miltenyi is providing these directly conjugated antibodies that are pre-labeled, but you can also now we use a kit from PG, what are they called? PG Biolabs or something, or
is it flexible?
Yeah. So exactly.
Flexible's really good.
Yeah. No, and that's actually super nice because what you can basically do is take any antibody. I think right now it's for mouse and rabbit, but you can take any antibody and label it, customly, and then put it on your samples. Which also opens up for using mouse antibodies in mouse, which is also super
and
nice.
A lot, at least with neuroscience, my background's immunology, so I don't know how I got here, but my background's immunology. So coming from a land of all the antibodies in every color of every type, neuroscience, the majority of antibodies are not conjugated. That's why suddenly getting directly conjugated antibodies is actually quite a big thing. And you're right. So from the point of view, that kit that you're talking about is really cool because it doesn't involve you putting your antibody on a filter. So if normal kits, I can't remember the name of, mix and stain is one of them. And a lot of them are like this. You have to put your antibody on a filter to wash out the liquid it was suspended in, and you can kind of concentrate your antibody again. But you have to recover it from this tiny membrane in like 10 microliters of liquid, and you're never going to get all of it back. So the flexible kit is just this plus this, wait 5 minutes, add this, 5 minutes, done, use. Perfect. And
it even works in the presence of carriers like PSA and stuff. So I think
it's really good.
Also an important thing.
And then your other options for that are you can either pre-conjugate with a fab fragment. That's something I tried a while ago. And nanobodies. And these are things that I'll be trying in the future. But it's all about ease and getting yourself to having a directly conjugated primary. It will say just if you can do that, if you have the chemistry know-how to just do that, do it. It will save you so much time.
So we always, for a long time, we have recommended using the alpha-smooth muscle actin antibody from Thermo Fisher as your internal control because it labels the large arteries in the tissue, which is always there, and it works in all species. So we always just add that in the beginning when we are testing new tissues or organs and stuff, just to have it. And then it shows kind of how well you have penetration. So
it's a good idea because
And that was the first primary antibody that we ever found that was already pre-conjugated.
Wow.
So we've been using that for a long time.
But it's true, one of the things that you have to consider when doing full anything, brain or organ, is is your antibody getting all the way through or is the target just not there? So at least with something like the smooth
Exactly.
muscle,
Yeah.
you know that protocol is enough to get an antibody all the way through. So at least you've got your time sorted. And that's that variable gone. It is a very useful thing to do.
So just here, if to wrap up a little bit, so how do you see this? How are you going to plan on using the TH staining going forward? Like, what is the biological question you would like to address with the TH staining?
Well, for now, because there's essentially these very, I'm not a neuroscientist. There's very specialized animal models that have sort of degradation of the pathway in slightly different areas at slightly different times. At the moment, this group just wants to look at the time course of how are the different areas of the brain in these different animals, how do they compare at different parts of the sort of degeneration pathway or degeneration cycle. And I guess in a way, this is the lovely part where I get to get it to work and then I give it away. So I have a working tool. I've shown them how to do it. They can get lovely resolution and the imaging and stuff. And other than that, it's on them to play. It's kind of the nice thing about my job is I get to do the interesting part of developing a thing and then package it up and away it goes and I do something else.
So obviously, I think when talking about Parkinson, one like the pathology behind the Parkinson is usually alpha-synuclein, right? Have you tried combining TH with alpha-synuclein?
Not yet. There is one of the things that I am keen. When I have time. I got, I think it was a neurofilament antibody as well, because, so when you're doing the full brain labeling, you're trying to get the concentration low enough to avoid this non-specific kind of rim around the outside, where the antibodies that's when the antibody concentration is too high and it blocks itself getting in. It's not specific, it's just an annoying fluorescent rim. So you have to make your concentration low enough for that. But I was wondering, if you put two antibodies in at the same time, is the total concentration of antibody actually something you have to consider? Or once you get one sorted, there's actually no issue to put a second one in. So the other antibody I'm going to use is neurofilament, because again, on the Miltenyi website, I found one that was proven for iDISCO. It's got a concentration that works. And I'll throw those in at the same time to see if it works. And then I guess once that is answered, then we can start being a bit fancier by throwing in different colors. And those colors being based around your standard 4, but light sheets don't like blue. So it's actually green, red, and far red.
Yes.
So yes, once I've tested this thing for my theory, it would be open to whatever targets they want. There would have to be an antibody that's proven to work, but it's early doors, but it's cool.
Yeah, and I think I mean obviously the whole TH pathway in Parkinson is kind of the main readout for the pathology that happens in response to the to the spreading of disease.
Lower TH, lower dopamine producing cells. It's it's quite a nice one to look at in that particular pathway. And then it can be advanced as well by throwing in some dyes. Dyes are also quite easy to get through a full brain with the right buffers. And then you're just chopping and changing all these different tools that you want to look at in the full brain, nice and easy. It's just good. It's just cool.
Well, and I think on that note, I mean, we could maybe start here
just
and just say whole brain is cool. Yeah, exactly. So, but thank you so much, Peter, for taking the time. And to anyone who's interested in watching the map of the month, they can go to Vibrant's webpage and have a look.
Yes.
And play around. So, thanks,
No
Peter.
problem.
Thanks for taking the time.
Thank you.
Bye-bye.